And today I tell you about next large section in physiology. We will discuss physiology of high nervous system. It's a large topic, and we will continue this topic during few next lectures. And first of all, you should know. I remind you. I hope you know this from anatomy. That our neuron system, our neural tissue in whole our body, divide on two parts: central neuron system and peripheral neuron system. Central neuron system, it's our brain and spinal cord, and peripheral neuron system, it's nerves and neuron ganglions. But common for both central and peripheral neuron system, they consist from neural cells. Previously, we tell about these cells. It's one of excitable cells in our body. And main role of these cells is providing action potential, transmitting from receptors to muscles to glands, transmitting signals. Nerves in periphery consist from several tissues. It's not only neural cells. You may see in this slide neural cells placed together and covering by connective tissue like fast cells, and together with neurons cells in nerves, there are vessels take place for realizing blood supply. And these many neuron cells, together with connective tissue, with blood vessels, produced nerves, which, like cable, consist from many isolated neural cells. Common for neural cells in our central and peripheral neuron system, they including several components. Neural cells may be different, you may find in this picture. They have different form, different size, but most typical for neural cells are large branches around the central part, around the soma of neuron cell. We have some branches, and these branches divide on two groups: axons and dendrites. Usually, we tell about neuron cells which have only one axon and many dendrites. But you may see it's not absolute. In some cases, we may find branches look near the same, many branches, only one large branch. That's why many students confused what's this axons, what's this dendrites. Actually, in physiology, we have good definition for this. Axons, it's those branches which send impulse away from neural body and transmit signals to other neurons. Dendrites, it's branches which send information from other neurons to neural body. That's why mostly we divide these branches not by size, not by placement, not by number, but by physiological functions. Dendrites acquiring information from environment, and axons send information to other cells. Additionally, in our neuron system, as central as peripheral, we have many supply cells. Usually, we named glial cells. It's common name for all supply cells, and this from English word glue. Glue may attach something to other particles and give ability to fixate. And glial cells like glue fixate neural cells in space and environment and give ability to form some tridimensional structure. But in nowadays, we tell about additional functions of glial cells. For example, we have astrocytes. It's one type of glial cells which usually placed between vessels, small vessels, and neural cells. And astrocytes realize immunic function. They help to regulate permeability of vessels, regulate blood supply, and regulate some protective functions. They block ability to passing toxins, some poisons to our neuron cells, and block our own immune system like leukocytes to came to our neuron cells. We have oligodendrocytes which form myelin sheet around the neurons. If you remember previously, I told you about other example of these cells, Schwann cells, which form myelin sheet. And we have microglial cells, which realize regulatory function to help provide normalized extracellular fluid components. And these microglial cells in nowadays are highly important for our central nervous system activity, for growing our brain and spinal cord, for placement and normal dividing of neurons. That's why we have many types of glial cells, and common for all these cells, they help for our neurons to normal activity of the cells. We have many classifications of our neuron system. First of all, like I previously told you, it's central and peripheral. Central is brain and spinal cord, peripheral it's neurons and ganglions. Ganglions, it's bodies of neurons which placed not in central neuron system. Because nerves normally it's axons, and nerves don't have soma, don't have body of neurons. That's why in peripheral neuron system we have ganglions and nerves. But we have additional functional classifications. You may see in this slide. We divide by direction of propagation impulses into afferent and efferent subdivisions of peripheral neuron system. What it means? Afferent subdivision, it's those nerves and ganglions which send impulses to our central neuron system from periphery, from receptors to brain and to spinal cord. Efferent subdivision of peripheral neuron system, it's those nerves which send impulses from brain or spinal cord to our muscles. We say about motor neurons. To our inner organs, we tell about autonomic neuron system with subdivision, sympathetic, parasympathetic neuron system, and to our glands. All these functions realized with help of efferent peripheral neuron system. For better understanding, we combine all components in one arch, which named reflectory arch. Because usually in our body, afferent, efferent placements connect together with help of central neuron system, and we may see like this arch. Impulses came to receptors. From receptors, with help of afferent neuron, came from periphery to our brain or spinal cord. In this brain or spinal cord, we have interneurons. It may be one, two, or many hundreds interneurons. It's not so important now. And interneurons connect with motor neurons, which send impulses away from central neuron system to our muscles, to our inner organs, to our glands. That's why for normal realized sum functions, impulse should traveling through all these components, from receptor to a working organ, muscle or gland. And this is highly important because it's normal pathway for realized neuron activity. This pathway usually named reflex or reflectory arc. More correct, tell about reflex like function. It's a response on some stimulus realized with reflectory arc. And reflectory arc, it's a pathway which including three main components like I previously showed you. Afferent pathway, intermittent, and efferent pathway. And these principles of reflectory arc, which lay in base of all neuron regulations, all types of neural activity, highly important in clinic, in medicine. Because if something goes wrong and this reflectory arc breaking down due to damage, for example, impulses didn't came through all these components, our neuron system don't realize the functions. Our muscles don't start contract. But it's not important on which stage reflectory arc was broken. It may be broken in in sensory part. It may be broken in central part, in efferent part, but a result will be same. Impulses didn't came from receptor to working organ. In clinic, we may check integrity of reflectory arc for some reflexes, for some types of responses. Most well known is knee-jerk reflex. If you came to neurologist, they will beating you by your knee with hammer, and it's test for checking integrity for reflectory arc, which realized knee-jerk reflex. So reflex its function, and for realize this function, we have a reflectory arc. This arc start from receptors, which placed in quadriceps muscle and in tendons from this quadriceps muscle. Impulses activate when tendons stretch quadriceps muscle and send impulses to our spinal cord. From spinal cord, efferent neuron send impulses backward to quadriceps muscle, and muscles start contraction. This realized integrity of knee-jerk reflex, and this reflex have protective function because our body don't know what starts stretching this muscle. Maybe it's hammer, but maybe it's some dangerous, and start involuntary protection from this stretching with contraction of quadriceps muscle. And if doctor, when they beating with hammer your knee, see that your legs start move, uh, doctor decide that this reflectory arc normal and there is no disorders on all these pathways. If a reaction is absent, it may be due to many uh, reasons with damaging sensory neuron, motor neuron, receptors. It's a common situation, but a doctor will start search to find what's reason for these disorders. That's why in neurological practice and clinic, we use many types of tests for checking integrity of neural pathways of reflectory arcs. It's only one example, but actually we have many reflectory arcs for innervate all our muscles in our legs, in our arms, in our inner organs. And some of these reflectory arcs we may test, we may check with neurological tests. And it's highly important for early uh, making proper diagnosis. Okay. Next, we will tell about some specific properties for working neurons together, because our neuron system, it's not only one neuron, it's many neurons. Usually we tell about total amount of neurons in our brain and spinal cord, like 10 power 100. It's huge amount of neurons, more than stars on the sky. And this huge amount of neurons should have some principles for uh, coordination, for combining the activity. For example, one principle may be principle of summation. You may see in this picture when several neurons combine and send impulses in one place. Signals from these several neurons may summarize, and if every stimulus is quite weak, subthreshold, only one stimulus, one neuron don't provide action potential, but many neurons together lead to summation and provide action potential. It's principles of summation. Summation may be spatial, when many neurons came together in one place, or temporal, when one neuron, but with very high frequency, send impulses through the synapse to other neuron or muscle or gland. And in this case, temporal summation lead to uh, depolarization on postsynaptic cell membrane. And in this postsynaptic cell membrane, like in this picture, you may see depolarization reach threshold, and as a result, action potential may start. And these principles are highly important for regulation our neuron system activity. For example, many everyday types of signals is very weak and they don't lead to some responses. But if it's many time repeating signals or combining signals from different places, they may lead to activation our neuron system. This way for change our behavior, to change our regulation, and even daily activity may be example of this regulation. For example, you're preparing to classes. If you read some text or lecture only once, usually this information will be sub-threshold. But if you read many times or you provide information from different sources, you read and spoke in this moment text which you read or try to write some notes. It's summarized information and they easily came to your brain and storing for the future. It's like example, of course, but it may be help you understand that these principles can coordinate our activity and increased response or vice versa decreased. And now I tell you about opposite situation, about decreasing response from some stimulus in our neuron system. Our neuron system have a very specific type of response which named inhibition. And inhibition is process which opposite to activation to production of action potential. Because inhibition it's temporal decreasing ability for producing action potential. And our neuron system have ability to decrease this action potential producing with many ways. Most common it's primary and secondary inhibition. You may see primary inhibition may be with different subtypes like presynaptic, postsynaptic. I tell you about this a bit later. But common for this group primary inhibition, it's inhibition with specific structures, some interconnections, synapses between neurons have specific components like inhibitory neurotransmitters. And if neurons have inhibitory neurotransmitters, they may realize primary inhibition. But in some cases, neurons have no specific structure, specific components, specific neurotransmitters for inhibition, but only type of signals or frequency of signals may lead to inhibition. And this type of inhibition without specific structures named secondary inhibition. Now we will tell about primary inhibition, and for better understanding you may see scans for this primary inhibition. On scans you may find a relationship between many neurons. You see it's two types of neurons. Black color, it's ordinary neurons which send impulses like action potential from one neuron to other through the synapses. But this propagation of impulses may regulate with inhibitory neurons. In this case, it's color neurons and this inhibitory neuron may have interconnection with excitatory neurons before synapse, before this part, synapse, and in this case we tell about presynaptic inhibition. Or second way, inhibitory neuron may attach to second neuron in chain to postsynaptic area, and we tell about postsynaptic inhibition. It's important because these two types of inhibition have different mechanisms, and in clinic, in practice, we may regulate these two types of inhibition with different ways. But common for all types of primary inhibition, they was discovered by Russian physiologist Ilya Sechenov. He was first who find inhibitory processes in brain, firstly frog brain, but in nowadays we find similar processes in human's brain. And we may say about balance in our central neuron system between two processes, activation, like action potential, and inhibition. And normally these two processes should be balanced. Normally we need balance between intensity of activatory processes and intensity of inhibitory processes. If these two processes is balanced, it's normal. But you may see imbalance may be very dangerous. For example, if excitatory processes prevalence and there is no inhibition in our brain, action potentials may increase our brain activity more and more and more, and lead to uncontrolled muscle contractions and loss of consciousness. One of example, it's epilepsy, seizures. Vice versa, imbalanced inhibitory processes may lead to coma and even may lead to death, because if all impulses to our muscles, to our heart, to our inner organs will be stopped, it may lead to stop our body activity and may lead to death. That's why normally we need balance between two processes, excitatory and inhibitory. I say about different mechanisms, and now we will see in this slide mechanism of postsynaptic inhibition. It's primary inhibition. It means this type of inhibition require specific inhibitory neurotransmitters. Usually, it's only one from neurotransmitters, gamma-aminobutyric acid or glycine. Common for postsynaptic inhibition, I previously told you, inhibitory neuron, which releasing GABA or glycine in environment, connect to postsynaptic cell membrane after synaptic cleft. And this postsynaptic inhibition changed activity of secondary neuron and decreased ability for provide action potential in this secondary neuron. Usually, postsynaptic inhibition realized with help of hyperpolarization, because glycine, for example, lead to open chlorine channels and entering chlorine ions in the cell. As a result, in the cell became more negative charge compared to outside of the cell. And this lead to hyperpolarization and decreasing excitability. You may see in this slide, it's opposite to depolarization to excitatory processes. Excitatory processes lead to decreasing charge between cell membrane and environment, and hyperpolarization increasing charge between intracellular and extracellular fluid. But this hyperpolarization, it's temporal process. After a short period of time, chlorine gates are closed and hyperpolarization disappear, and neuron cells are ready to react on some stimulus again. But during this hyperpolarization time, neurons may be blocked and not react. Second type of primary inhibition, it's presynaptic. This type of inhibition realized only with help of gamma-aminobutyric acid and lead to depolarization. And it may be like a paradox. If you listen in me, you may remember that 2 minutes ago I say depolarization, it's excitatory process. But with help of presynaptic inhibition, depolarization lead to block propagation impulses from one neuron to other. And this may happen if depolarization start very slowly and last during long period of time. Presynaptic inhibition have more time compared to postsynaptic, and mechanism associated with accommodation. If you remember on previous week, I tell you about this process, and I tell that accommodation, it's changing sensitivity, it's decreasing sensitivity. With help of accommodation, our cells decreased on some period ability to respond on some stimulus. In presynaptic inhibition, we have first period when accommodation not start yet, and in this period excitability may increase, and this principle differ between presynaptic and postsynaptic inhibition. In presynaptic inhibition, action potential may traveling through this synapse from presynaptic to postsynaptic neuron in first period of inhibitory process when accommodation still not very strong. In postsynaptic inhibition, vice versa. In first moment of inhibition, action potential not came and no response happened. It's more stronger inhibition postsynaptic, but last during short period of time. Presynaptic not so strong, but can take place during long period of time. Both pre and postsynaptic inhibition may block excitatory neurons with different pathways. In these schemes, you may find main types of inhibition. For example, recurrent inhibition. What you may see in this slide. You may see that excitatory neuron send impulses to other neuron, but with help of branches, this excitatory neuron stimulate neighborhood inhibitory neuron, and inhibitory neuron with feedback block first excitatory neuron. In other name, in other word, you may say that neuron block themselves through inhibitory neuron, but as a result, this neuron block themselves. This mechanism highly important for regulation intensity of activity. All our skeletal muscles, for example, have recurrent inhibition, and this may be helpful for decreased time of muscle contraction. Our muscles don't contract during a long period of time. With help of recurrent inhibition, even if signals came to muscles, these signals will be blocked when neurons stop producing action potential and start blocking themselves. This gives time for muscles, time for recovering, time for relaxing. One minute, sorry. Sorry. Next one, reciprocal inhibition. And this type of inhibition, again, very useful in our muscles, but antagonistic muscles. For example, in your arms and legs, you have muscles which move in opposite directions, your arm or your leg. And you may bend some extremities, arm or leg, or you may extend extremity. But you could not use both group of muscles for contraction and for extension. Normally, only one type of activity. And even if some problems lead to stimulate both groups of neurons, motor neurons, one muscle not start contraction due to reciprocal inhibition. You may see in this picture when motor neurons send impulses to muscle through inhibitory neuron, they block activity of antagonistic muscle. We start, for example, contraction and we block muscles for extension, and vice versa. Next type is lateral inhibition. Lateral inhibition may be useful in situations when we need block many parallel pathways. It's very important and widely used in our eyeballs, in our retina. With help of lateral inhibition, visual signals filtrate decreased intensity of response, and only one neuron send impulse to our brain from retina in our eyeball, and many other neighborhood neurons will stop the activity. This lead to filtrate some information and increased ability for recognized vision to improve visual acuity. Now we tell about secondary inhibition or inhibition without neurotransmitters. First of all, it's pessimal inhibition. Pessimal, from term pessimal, it's very high frequency of stimulation when second stimulus came to refractory period from first stimulus. About these terms, I tell you about in first lecture. Was this refractory period? Was this action potential? And when second stimulus came during refractory period from first one, second stimulus not lead to response, not lead to produced action potential. That's why a very high frequency of stimulations lead to block second, third stimulus, and so on. First one provide response, provide action potential, but next will be blocked. It's not due to some specific neurotransmitters, but only due to properties of excitable tissues. Low all or none when action potential produced until action potential finished. No other stimulus will provide response. And inhibition after excitation. It's other situation, but again you might find on this graph when action potential finished in many cells and many tissues, they transfer to after potential, to hyperpolarization period. Again, I tell you about this period in first lecture. But hyperpolarization lead to decreased excitability. And if next stimulus came in this period from first one, second stimulus not lead to produce an action potential. We may say second stimulus block, inhibit. In this picture, you may find that different neurons in different parts of our body, in different parts of our brain, have different ability to after processes. And some components of our neuron system have very small after polarization. Mostly it's our motor neurons which send impulses to muscles and impulses which came from brain to spinal cord. But some neurons, especially it's neurons in thalamus, in cortex, have a very intensive after polarization, and these neurons regulate excitability with help of this pessimal inhibition, inhibition after excitation. During period of after polarization, these neurons could not provide next response. Additionally, our neuron system have ability for regulate intensity of inhibition with different amount of inhibitory neurotransmitters in primary inhibitory processes. You may find in this picture inhibitory processes may be increased or decreased with help of inhibitory neurotransmitters. Our pharmacologists know about inhibition and try to use this process in practice. For example, if you know about such diseases like seizures, we have many anti-epileptic drugs for curing seizures. And all these drugs stimulate inhibitory processes. What it means? They block action potentials or decreased frequency of action potentials produced. And as a result, in imbalanced brain, return balance between excitation and inhibition. Or some sedative drugs, sleeping pills, all these medicines again used mechanisms for increasing inhibition. Some of these drugs work only in primary neurons because many of them regulate releasing of GABA or efficiency of receptors to GABA, gamma-aminobutyric acid. And you may find in this slide many groups of medicines have ability to attach to receptors for GABA. It's anti-epileptic drugs, like I told you previously, benzodiazepines. It's sleeping pills, barbiturates, and even hormones like steroids. It's our own hormones and all drugs which work similar to our hormones. Increased permeability of cell membrane to GABA and increased efficiency of receptors to GABA. As a result, inhibitory processes increased and cell on some time became less excitable. Cell not react on stimulus, and in some cases it may be good. Cell have time for recovering, for return to normal state. That's why inhibitory processes are widely used and very popular in medicine. Different parts of our central neuron system have different prevalence between excitation and inhibition. But in general, we used common principles, principles of hierarchy in our central neuron system. What's this hierarchy? You might find in this picture levels of organization. In our central neuron system, brain and spinal cord, we have cerebral cortex, like top level for regulation. And signals from cerebral cortex came to neurons placed below cortex in subcortical regions, like basal ganglia, brainstem, cerebellum, and so on. From these regions, impulses came to next level, third level, it's spinal cord. And from spinal cord, through motor neurons, impulses came to working organs. It may be muscles or glands. So for normal activity, we used all these steps. But second principles, it's principles of feedback. And you may find in this slide, when our spinal cord sends impulses to muscles, from muscles, impulses return back to spinal cord because every muscle have receptors, stretching receptors, tension receptors. Uh, if this receptors activated, spinal cord receives signal that muscle realized their function. They contract and it's okay. We may stop stimulate muscle. This stopping realized with help of inhibitory neurons. So all feedback mechanisms associate with inhibition. And you may see many feedback mechanisms from muscles to spinal cord, from subcortical regions to cortex. All these feedback mechanisms realized with help of inhibition. And again, I remind you about importance of balance between inhibition and excitation. Because imbalanced processes may lead to death, as in situation when prevalence inhibition, as in opposite situation with prevalence excitation. It's abnormal and may lead to diseases. That's why in clinic, if we find this imbalance, we may use some drugs for stimulate inhibition or decreased inhibition, and it may be very good, it may be important for normal regulation and healing patient. And last topic for today, methods for research in our neuron system. We have quite a serious problem if we want to know more about functions of our brain, because brain covered by skull, and we could not touch our brain, see how this brain works. That's why for physiology of central neuron system, we used non-invasive methods. What it means? Methods which don't change integrity of our body, don't cut skin, don't breaking down skull, don't breaking down brain or spinal cord. And this method's quite popular in nowadays because patients didn't like when you break integrity of the tissues. And first, most popular method is EEG, electroencephalography. In Wikipedia and many modern sources of information, you may find that EEG was discovered by Hans Berger. It's Austrian scientist. But actually, more than 20 years before Hans Berger discovering this, Russian scientist Pravdich Neminsky find this EEG activity in animals. That's why EEG it's not so modern method. They came from 19th century, but became popular only in 20th century after Hans Berger discovered. And EEG, it's method for recording electromagnetic fields which produced during activity of neurons. Neurons which placed close to skull, it's cortical neurons, pyramidal neurons, and glial cells around the neurons. All these cells change permeability of cell membranes, produced electromagnetic fields, and our modern devices may measuring change of electromagnetic fields and record on paper or on computer for analyzing. On EEG, we may find different waves. It's four common waves you may see in this picture. They are named from slowest one delta, more fast waves 4 to 7 times per second. It's theta, 8 to 12 hertz. It's alpha waves, and more than 13 hertz. It's beta waves. Additionally, nowadays, we tell about other frequencies, gamma, epsilon waves, but if you not work like a neurologist, it's not important for you. Waves are important for describing integral activity of our brain, because if it's healthy brain at rest, we are mostly measuring alpha waves, and we're measuring these waves in occipital lobes of our brain. If we didn't find alpha waves, or they placed not in proper place, our brain works not as usual. It may be due to physiological reasons, exciting, or dreaming, or sleeping, or it may be pathological states like some inflammatory diseases, traumas, which break normal waves on EEG. Additionally, we may check quality of brain activity by alpha block, because it's interesting that alpha waves disappear when person open his eyes. When person closed eyes, alpha waves appear again. This demonstrate source of alpha waves. Usually it's waves which localized in our visual analyzer in occipital lobe of brain. We have neurons analyzed information from our eyeballs. If these neurons in relaxed state, they produce alpha activity. If these neurons overactive, they will produce beta activity and alpha waves disappear. And in some emotions, in children, and in many pathological states, we may find other waves. It's slow waves like theta and very slow like delta waves. So for doctors, even prevalence of waves, different frequencies, different position of waves, may be important for describe level of activity, functional state of brain. Patient in relaxed state or overactive patient in sleeping state or not, and some pathological states. Previously, I tell you about inflammations may lead to produced abnormal activity. Some tumors may change EEG. That's why it may be informative for find these disorders. And even changed electrolyte balance in blood, especially for sodium, for calcium ions. And as a result, this imbalance may change EEG and lead to placement pathological rhythms. But most well-known pathological state which discovering by EEG, it's epilepsy or seizures. And we may find localization of abnormal neurons in brain and find epilepsy with help of EEG. For example, I demonstrate you two pictures, two types of brain activity. And first one, it's a relaxed, healthy body. You may see it's alpha waves present in occipital lobe, and it's okay. And seizures in second picture, you may see how dramatically differ EEG on first and second slides. During seizures, we may find high amplitude waves, slow waves, or fast and slow waves together. And usually they localized only in only some one part of brain. It's localized abnormal activity, or may localized in whole our brain, in all electrodes. It's generalized abnormal activity. That's why EEG may be helpful for find source of pathological activity, where problems in this patient we may think even without open skull and touch the brain. That's why this method is popular in nowadays. And additional, these methods may be popular with many mathematical methods for localization activity, like in this picture you may find okay, sorry. Mathematical methods can help us easily recognize where in brain some kinds of activity take place. For example, when person in relaxed state, we might find increased activity in his visual cortex. And again, it's normal for healthy body, it's okay. In some seizures, again, with help of localization method, we may find where pathological state placed. And it may be informative when we combine several methods, EEG and MRI or PET methods. its structure methods which show structures of our brain, and EEG its functional method which show activity and prevalence between excitation and inhibition in brain. In these pictures, you may see combining of these two methods, and it's easily distinguished where pathological states take place. That's all what I want to say for today. Thank you. If you have any questions, you may ask me.